It is my pleasure and honor to introduce Brett Monia, a well-known individual, I think, in this society, so I don't have to tell you a whole lot, but I do want to tell you a few personal things. So, first of all, the way did I get the right one? Yeah. So, Brett Monia is, I think, has a legacy of innovation that exceeds many folks in this endeavor. He's a co-founder of Ionis, 1989. He's the current CEO of Ionis, has been since 2020. He's my boss. He's led ASO research at Ionis for over 20 years, and R&D scientist, innovator, inventor, over 100 patents, a real leader. Today, Brett is my boss, and if you buy me a drink, I can tell you a little bit more about my boss. He is also a founder and member of the initial board of directors here at OTS. He was a board member from 2003 to 2018, 8 consecutive terms. So I don't know if it was the longest, but it was close to the longest. He was the president in 2015, 2016, and the annual meeting event co-chair in 2010 and 2013. That's when I started getting involved, because he kind of said, hey, you need to be going to these meetings. So the annual meeting event committee, he was on this 2008, 2015, 2017, and of course he's on our scientific advisory council as well since 2019. A few pictures. I think this will bring back a few memories. Going from 2011, the 2013 Naples meeting, and just a note about the Naples meeting, I think that was the first meeting where a party was introduced. This was like kind of a dream of Brett's, and he wanted to really push for this. At the end of the meetings, we need to have a party. Well, the first party, I don't know if it was in Naples, I think it was kind of lame. Wine tasting, a few flutes. I don't know if everybody stayed awake through this one. And then in San Diego in 2014, and 2015 in Leiden, and Amico, of course, you can see in that shot. So the OTS annual meeting wrap-up party, I think this is Brett's legacy. Everybody loves a party, right? As of 2013, we had that lame 2013 wrap-up party. But even though it was tame, it was the start of a new dream. And then everybody came to San Diego, and we took everybody out on the ship. You remember the mariachis? Had a mariachi band. There's Brett giving a talk on the down under in the ship itself. And this was the one where Mossad got Brett to go on to the helicopter. Did you guys? Yeah. Almost killed him. So, but that's, everybody survived, and we're all still here today. And then the OTS annual meeting wrap-up party. I just want to say he had a real party set up, or he certainly encouraged a real party set up in 2015. There was going to be dancing, and there was. There was the board of director conga line dancing at this party, and Brett slept through it. Let's see what happened. Yeah, this was the conga line. Here's a little bit of dancing. Everybody got a little crazy. Okay. And then, sadly, Brett was exhausted. Well, in 2015, something very important was happening. The company, ISIS, was turning to a new name, Ionis, and Brett was in charge. Make it happen. And, well, he was in his hotel room making it happen and fell asleep. So here is the burying of ISIS. So this is a little personal example of what was happening as ISIS went down and Ionis rose. And so we are now Ionis, and Brett is our current CEO. So please enjoy this short video, followed by Brett himself, who will come out and present. Thank you.
I first met Brett in 1987. At the time, he was just starting as a graduate student. In the last 30-plus years that I've known Brett, my impression of just been reinforced that he is a hard worker, and he's been very productive as a scientist here. He's one of the few people that you could say have helped create oligonucleotide therapeutics.
Brett is like a great example how an extremely intelligent person with a lot of drive and belief in an idea, who's willing to stick with it for an amazing amount of time, can really do some like world-changing, healthcare related science.
The field really has gone from ground zero, frankly, 35 years ago, where Ionis, its predecessor company, was the only player, to now over 150 companies that focus on oligonucleotide therapeutics. Brett's leadership, his role in guiding the technology, its application to important clinical problems, has been at the forefront of moving that industry along for the benefit of patients.
One day he said to me, the pharmacokinetic data is showing that these oligonucleotides tend to gravitate to the liver. Why don't we work on medicines that you can create by dealing with the liver? It seems very obvious in hindsight, but none of the rest of us thought of it. And as a result, we started making medicines that did work. And now we have an entire portfolio based upon that insight.
I think one highlight for Brett's career is his contribution to the TTR amyloidosis and finding the therapy for that. So I think Brett really is a pioneer in finding this disease-modifying therapies and bring them to the patients.
Brett's contribution to the antisense oligonucleotide field has been incredible. Ionis has been one of the leading companies in the world in this space. He's run research, became head of R&D, now he's CEO of the company, and he's obviously been at the forefront of this technology and all it's delivered over many years, and it's really exciting to start to see these platforms become like small molecular antibody platforms and really turning, I think, a great innovation, a great science into something that's making a tangible difference to patients and really turning science into medicine.
Brett was one of the founding members of the OTS and has been in various leadership positions since its founding. And it's really a great honor for him to receive this, in particular since he's contributed so much to the organization.
The first time I met with Brett when he was president, my colleague Jerry and I met with him, and this is what he showed up with, which was a list of all of the goals he wanted to accomplish and the action items that we needed to take to get those things done. During Brett's time as president, we accomplished everything on this list and then some.
Brett built OTS. He knew whom to bring it to the table so that he will form a wonderful leadership. So which started with a few hundred people. Now thousands of people are attending Oligonucleotide Therapeutics Society meeting now. And such a growth is because of Brett's early days vision in building this society.
The impact that he has on up-and-coming leaders and up-and-coming scientists and business people is just really profound. His humble nature makes him very approachable, and I think then people listen to his advice, and I think he's had a huge impact on people.
He's not hierarchical. He will listen to anybody at any level who's got something that's really good to say. Uses really good listening skills and lets people complete like what their thoughts are and share them openly. I have to think that that actually contributed to how his scientists were able to innovate, those that reported to him.
To me, he epitomizes sort of what leadership is, which is it's science, it's business, it's finance, it's regulatory, it's this and that. And you have to put great teams of people together. And I think Brett's got the right personality for that, because one, he's just a great leader.
It's a tough business that we're in. Lots of pitfalls, and Brett has a way of rallying everyone together without making a big crisis out of everything and bringing people together so that they can get the really hard tasks done.
I think what you see in Brett, which is a little bit unusual in a PhD nerd, is an MD heart. He has the heart for the patients, and that's always been foremost at this company, but it's always been foremost with Brett.
It's remarkable and inspiring to see the work that's been done impacting patients directly. I've seen it work, and I've seen it happen for the patient community, and to know where it started, world-class activity and research led by Brett here is amazing.
You know, as somebody who went to medical school myself and has been involved in the medical field, you know, I understand that at the end of the line here is what it means to patients, but had never really fully thought through not just what it means to patients, but what it means to, you know, physicians, that somebody is doing the painstaking work. Brett, I hope Ionis does a lot more of that, but even if we've only done it for some patients and some diseases and some clinicians, man, what else in life is worth doing that's better than that?
I don't think that anyone in the world has had the influence that Brett has had in terms of putting important medicines into the clinic to change the progression of neurodegenerative diseases.
I'm really happy that Brett now receives this Lifetime Achievement Award. I think it's a bit overdue. In hindsight, we might realize that he's done a lot more than we know about.
Good morning, everybody. I am obviously thrilled and incredibly honored and humbled to receive this award from the OTS, a society that you heard about earlier, how much I've been involved with and how much tremendous respect I have for this society. Thank you, Richard, for the kind words, and thank you for the very nice video. A lot of pressure to deliver on all that people said I've done in my career, but I'll do my best. You know, 35 years is a long time, and therefore there's a lot to cover. So I better get started here, and I'm going to come back to the acknowledgments at the end of my presentation. So I was thinking several months ago about what I should talk about at this presentation, and I asked myself, you know, what should I focus on? Like I just said, should I focus on the past or the present, disappointments and learnings from those disappointments, successes, lessons learned? And what I decided was I'm going to touch on all that, the journey, the 35-year journey of my career that took me to where I am today as the CEO of Ionis. And I'm going to really, really try not to bore you all to tears. I'm going to try to keep it upbeat and move it, and I'm going to move quickly here. So let me start with the agenda. I view this as chapters, if you will. I'm going to start with the early years in my career, and then I'm going to move into a second chapter called Making Lemonade Out of Lemons, and I'll let your curiosity get the best of you for that, and I'll hold that till we get there. The third is benches to bedsides. Fourth is a new job, a new plan, and then I'll close with some closing thoughts, reflections. So, early years. I gotta start with some backdrop, right? Let's provide a foundation here. When this whole field of antisense oligonucleotides began, we were 10 to 15 years into the biotech explosion, the biotech era, if you will, the mid-'70s to through the 80s. It was a scientific and medical revolution. It was incredibly exciting. Novel therapeutics were being conceived. A lot of new biotechs were starting up. Many of the names that I've listed here in this era. A lot was going on. And it was desperately needed. It was desperately needed. Biotech was the answer to the inefficiencies that large pharma was creating. We weren't tackling chronic diseases effectively enough, not nearly enough. Sorry to the pharma folks. You guys are great. But biotech really was viewed as the answer to solving problems for human therapeutics, and it was a really exciting time. Based on recombinant DNA technology, based on DNA sequencing, based on the emergence of the monoclonal antibody platforms. And it was around this time that work by Paul Zemechnik and many others came up with a crazy idea that maybe we can create a new platform for drug discovery called antisense. And I purposely used the most simple version of antisense that I could possibly find. I could certainly have shown you all kinds of mechanisms of action and all that stuff. But I wanted to show you this simple version because that's what we knew about antisense in the mid to late 80s when we were getting started. We didn't know anything about whether these drugs would work, how to modify them, give them drug-like properties, distribution, toxicology, manufacturing. I could go on and on and on. We knew absolutely nothing, but the concept was elegant. And you all know the concept. This is the OTS, so I don't have to go into that. So, 1989, biotech is roaring. The antisense concept is driving tremendous excitement as the answer to many of our problems with respect to human diseases. And companies were founded, and I list them here. You could read them. I don't need to read them all. What I can tell you is that ISIS, now Ionis, and I'll try not to go back and forth too much. I'll try to say Ionis going forward, and ABI Biopharma, which is now Sarepta, were the only two that stuck with it from the beginning. Others failed or changed the direction. They moved away from antisense either to related fields or some entirely different fields. But we persevered, and we began, and that's when I began, as Richard mentioned. This is me when I came out of graduate school to help start Ionis in 1989. I was recruited from graduate school. And I was a molecular pharmacologist, pursuing an MD degree at the time as well. And I put my MD on hold. I figured I could always go to medical school, but when will I get an opportunity to actually potentially create a new paradigm and build a new company for human therapeutics? So maybe I'll go to medical school again. I'll go back if they'll have me. But I put it on hold, got my PhD, didn't do a postdoc. I was trained, and I was brought over to help start the company. And I focused on specificity of antisense drugs, mechanisms of action, and I did a great deal of work working with our outstanding medicinal chemistry team out at Ionis to identify the best possible chemistries, to give them the best possible drug-like properties to make them work in vivo, in humans. And I published a lot. I'm not going to go through all these publications, but a lot of early research focused on medicinal chemistry, working with great chemists like Dan Cook and Mano Manoharan, Eric Swayze, Rich Griffey, and Henry Sassmore, and others. And also mechanisms of action and specificity. We focused on the RAS oncogene and showed for the first time that you can actually get single-base specificity with an antisense oligonucleotide, if you can get it into cells, of course. I just want to touch on one part of this research that I led with others, my colleagues, of course, at Ionis at the time, which was mechanisms of action and the work we did with RNase-H. And you can read the title of this JBC publication back in 1993. And as I said, we were doing, me and my team were doing extensive work with the medicinal chemists at Ionis at the time, focused on backbone modifications, heterocycle modifications, sugar modifications, and we really thought that the sugar modifications, like 2'-O-methyl, 2'-O-propyl, other 2' modifications, were really going to deliver and help us increase potency, stability, and other aspects. But what we also recognized was that these chemistries weren't working when they're fully modified and introduced into cells. And at this time, I need to also tell you that the mechanisms of action that we know antisense oligonucleotides work today were not generally well well embraced at the time. In fact, most people believed that an antisense oligonucleotide was going to block protein translation by blocking translation, by targeting the AUG or that, or upstream of the AUG and block translation, and little credence was given to a destabilization mechanism of action like RNase-H. This has completely changed now, of course. So in working with my chemistry team, one of the things I asked simply was, well, these 2' modifications aren't working. Well, what if we slowly remove the 2' modifications from the antisense oligonucleotide? Seems pretty obvious, right? But not many people were doing it. Nobody that I know was doing it at the time. And this is a SAR that we did, a simple SAR we did in which we removed the 2' methyl modifications selectively a couple of bases at a time. And then I did an in vitro RNase-H assay. And what you see here is the cleavage products is that once we got to a gap of 5 bases or so, deoxyphosphorothioate gap, that we can get RNase-H cleavage, and it got a little better if you opened up the gap a little bit more. And then what was really exciting was, and I remember exactly where I was in the lab when I revealed this data, was that it correlated perfectly with antisense activity in cells in vitro that are in which the oligonucleotides are transfected in. You can see the activity of the 5-base, 7-base, and 9-base gap, and it correlates perfectly with the activation of RNase-H in vitro. And in fact, it even suggested, if you look at the deoxy oligonucleotide, that these high-affinity 2' methyl modifications would be even more potent. And in fact, we demonstrated exactly that. Using these gapmer approaches, we actually showed that if you can increase affinity, there's a direct correlation with potency. So you can see that the higher the TM, the lower the IC50, and that was really remarkable, and very it was very pleasing. At the same time, we were then asking the question of how can I was asking the question with the team, how can we approach this in vivo, not just in cell culture, right? And at the time, the whole field was focused on oncology almost. I shouldn't say the whole field, but it was pretty much dominated by cancer applications, tumor xenografts, orthotopic cancer models, and so on. And it was very difficult to reproduce. It wasn't a fast-moving way to screen medicinal chemistries. And a few benches away from me, when I was in the lab, a fellow named Nick Dean and Rob McKay, his technician, Rob McKay, were doing some work with simple first-generation phosphorothioate oligodeoxynucleotides, and they were injecting them into mice. And they were just asking the question, does it work? Can Brett's RNase-H mechanism actually work in vivo? And it was astonishing that what they found in that publication, which gets absolutely no attention. That certainly doesn't get the attention it deserves, this publication. And for you youngsters out there, this is a northern blot. This is looking at RNA going down. PKC alpha is the target. It's not a particularly good northern blot, but it's a northern blot. And what they showed was that if you just injected these things into animals, you get activity in the liver. You get activity in the liver, and then we showed in the hepatocyte using cellular fractionation methods and stuff like that. It was very specific. It doesn't affect the other isoforms or so. So I asked the question, well, why don't we just use that as our SAR and evaluate all the different chemistries that we were considering to be the chemistry of choice for our platform going forward? And that's exactly what we did. This is just one example of many hundreds of experiments I did with my team to look at different chemistries and so on. And this just shows the 2' Mo, 2' methoxyethyl chemistry versus 2' methyl versus first-generation deoxy, showing a dose response in mouse liver. And the duration on the right side of the slide, showing it's more durable. The 2' Mo is more durable and it's more potent. And this is a northern blot that I did, which is really good. You can see the target is RAF kinase. And we're getting very nice knockdown of RAF with the 2' Mo chemistry. So what this led to, I mean, I'm really summarizing years and years of work, was the selection of the most commonly used chemical modification in antisense drugs today, using the design that we worked out at Ionis at the time, which is the gapmer approach. The 2' methoxyethyl chimeric ASO, or gapmer, that we also often refer to. And we move forward with this chemistry going forward. Now, remember the liver work that I just took you through, okay? Great for SAR, but it ended up being a lot more. So now I'd like to move into my second chapter, making lemonade out of lemons. As I said earlier, most of the field at that time was focused on cancer. People believed that antisense drugs were going to be the magic bullet for oncology. All the oncogenes were being identified, and the specificity that antisense offered, people believed that it was going to solve cancer. And everybody was working xenografts, and including me. I was responsible for most of our oncology drug discovery work at the time at Ionis. And we worked for years on this, and we put forward molecules, and we had a partnership. And it was a dark time. Something happened. As I mentioned, in this time, oncology was the focus of antisense drug discovery. It was difficult to reproduce. A lot of people didn't believe it. A lot of trouble was starting. And Ionis was entirely dependent on a pharma to do xenograft, all their cancer animal work. We didn't even have an animal facility at Ionis at the time. And we had 2 drugs that went through our partner, moved into clinical testing, and was in patients. And I remember exactly the day and the time and where I was when I got the call from a senior executive at our pharma partner, in which I was informed by a senior executive that essentially all the data, in vivo data, was fraud, was made up. And in fact, the investigator that was responsible for it admitted it, and he was let go. And it wasn't just the antisense programs at our pharma partner. It was also their small molecule programs and so on. There was a big presentation at ASCO, apologizing, blah, blah, blah. And we had to do a lot of things, like retractions, investigations, and so forth and so on. But what this made me do, besides, as I just told you, was I was responsible for at least a lot of the oncology research at Ionis at the time. And it made me wonder a few things. Where do we go from here? Is there convincing antisense activity that we can rely on in animals? And if so, where, and can we exploit it for drug discovery? And that's when you get it. You know where I'm going. I harkened back to the liver. And I am not a hepatologist. I mean, I am today, I believe, but because of everything I know, but I had to study and pour myself over books and publications on what does the liver do. Because I got all this reproducible data when I was doing all this medicinal chemistry SAR, and I asked, well, can we do drug discovery in the liver? Folks, nobody was working on the liver at the time, as was mentioned by Dave Becker in that opening video. And I mean, holy crap, the liver does a lot. I mean, I couldn't believe everything that the liver did, the proteins that it produced that affect the immune system, detoxification, nutrients, macronutrients, risk factors that came out of the liver, blood sugar balance, coagulation factors, and so on. Now, at this time, I was moving into my role as head of drug discovery. I shouldn't say head. I created the drug discovery group at Ionis, and I kind of forced myself into being the leader of drug discovery at Ionis, which then I really focused the organization on the liver. We were still doing other things, of course, and neurology was just starting. We were just starting to do neurology at Ionis, but we focused the liver, and I created a cardiovascular group, an exploratory drug discovery group, and a metabolic disease group focused on these 3 areas: metabolic disease, cardiovascular, and so on. We were incredibly productive. We uncovered so much new biology. We focused initially on diabetes, type 2 diabetes, right? And these are papers that I published with my team and with really, really smart and well-regarded experts in academia that were experts in insulin sensitivity, glucose production, hepatic fat mechanisms of action, and so forth. We published a ton on this area, and it was incredibly productive. And we moved some drugs into clinical development, but our chemistry platform was not yet ready for chronic asymptomatic diseases like type 2 diabetes, right? Wasn't quite ready yet. This is before GalNAc. So these were higher doses and frequent dosing. But we uncovered a lot of great new biology, and it really provided a resurgence if you will, in the antisense field, at least at Ionis, and I know with a lot of collaborators, that then came back to the table and said, we want to partner with you on metabolic diseases. And although none of the drugs that came from our diabetes efforts at the time made it to patients, ultimately, FDA approval, it reinvigorated the antisense field, certainly in my community. I can't speak for all. And it allowed us to fund the company, and it reemerged from a time that was very difficult in the antisense field, and people were very much distraught with the inability to deliver on cancer applications. These are, in just a very short period of time, we did a lot of partnerships, and I led the creation of these partnerships at the time, and it really helped fund the company and kept us moving forward. I want to tell you one other story before moving on to the next section, which is additional work that my exploratory disease group did. So while we were doing the diabetes research, we were working on cardiovascular applications and antithrombotic applications. And the thrombosis research was very, very interesting. And this was work that I initiated with my team, trying to identify the optimal antithrombotic agent to prevent blood clots without causing bleeding, right? That's the big issue with antithrombotic drugs today, is that you can block clots from forming, but you expose patients to bleed. It's like a seesaw. You can't get both. You can't prevent clot formation and not cause bleeding. However, I was doing work with, I was talking to an investigator in the Netherlands named Eric Stroes at the time. And Eric pointed me to a paper that showed that people that are deficient in factor 11 in the intrinsic coagulation pathway have a much lower risk of cardiovascular disease, to stroke, heart attacks, and so on, and they don't bleed. And they're deficient in a coagulation factor. So we pursued factor 11 to try to get to that holy grail within the intrinsic coagulation pathway. And this is work I did with Hong Zhang, Jeff Crosby, Rob McLeod, and many others, of course, in the laboratory, in which we showed, this is an RNase protection assay, folks, this is in the northern blot, in which we showed that we can knock down factor 11, shown there in the factor 11 column, very effectively at the RNA level and at the protein level. Very specifically, we also looked at other coagulation factors, but we really focused on factor 11, and what we showed in animal models of thrombosis and all kinds of animal models of thrombosis was that we can, as shown in the bottom right corner, we can knock down thrombosis, prevent thrombosis from happening in a dose-responsive manner, and shown in the black line, not cause bleeding. This was the holy grail in the whole field. And compared to the state-of-the-art inhibitors at that time, a factor 10A inhibitor, just like we see in humans, causes bleeding, as does more traditional approaches like Coumadin or low molecular weight heparin. So we moved this program into clinical development. Still, nobody bought it. People did not think you can achieve both antithrombotic effects without causing bleeding. We brought it into phase 1 development. This is work I did with Sanjay Banat. And the first proof of concepts phase 2 study, the knockdown of factor 11, there's normal volunteers. Then we moved into proof of concept in patients undergoing arthroscopic knee surgery, so knee replacement. These people are prone for thrombosis and, of course, bleeding. And what we showed was that we can have an even greater antithrombotic effect by targeting factor 11 in this setting, better than the low molecular weight heparin and enoxaparin, and have a greatly reduced risk of bleeding. We proved the concept that you can differentiate bleeding from thrombosis. And this reinvigorated the field in antithrombotic research to focus on, well, maybe we should go after factor 11 because there really was no novelty that was coming forward beyond the factor 10A inhibitors that were coming at the time. And we went further. We not only showed the effects in knee replacement, we showed the effects in end-stage renal disease. End-stage renal disease patients are highly prone to thrombosis, and they're highly prone to bleeding. And what we showed was that we can block thrombosis in these patients and not cause bleeding. We published this, and at that time, this really got people interested. We did a partnership with a pharmaceutical company. I won't know the name. And unfortunately, we were 100% reliant on them to do late-stage development at the time, as well as to bring a product to patients, commercialize the drugs. And what they ended up doing was to put ours on the back burner and develop their own program internally. And that program is now in phase 3 development. I'll let you decide which company that is. But as I said earlier, this did reinvigorate the whole antithrombosis field. And today, based on the validation of factor 11 that we did with our first antisense approaches, which was again a non-GalNAc, new programs have now reached phase 3 development using monoclonal antibodies or small molecules. Now, have no fear, folks. There's a happy ending to this story. Unfortunately, our factor 11, which I still believe is the most effective and produces the most consistent effects compared to these other agents, did not make it to the finish line. But we uncovered new biology, and I believe that that's going to help a lot of patients long term. But what's really exciting is in parallel with all of this work on diabetes and thrombosis that we were doing in my drug discovery group at the time, we created an amazing pipeline of liver targets. Cardiovascular disease, you know many of these targets. Ionis was first. Hypertension, hemostasis, diabetes, as I mentioned already, and other indications like kidney diseases, and of course, amyloidosis. And that pipeline grew and was a top priority for me and became a top priority for Ionis. So now moving on to the next chapter. I forgot to mention earlier why I went to college and graduate school. I went to college and graduate school because I wanted to discover and bring forward new medicines for devastating diseases. It was always my area of priority in my academic life. And that's why I came to Ionis too, of course, is because I felt that maybe we could do something really different than nobody's ever done before. During my time in my career at Ionis, I had the opportunity and the privilege to have had my hand directly involved in shepherding drug discovery ideas from concept, through research, through development, and to patients, benches to bedsides. And I am incredibly proud of that. That's why I got into this business. And I want to give you 2 stories about my benches to bedside stories. There's several, but I obviously have limited time. And I have to start with transthyretin TTR amyloidosis, Shu-Ling mentioned in the video earlier today. So I think you're all familiar with this. This is a great win for the oligonucleotide therapeutics field. I'm sorry, I got to go back. So what is transthyretin amyloidosis? It's caused by a protein that's produced in the liver called transthyretin. It's a tetrameric protein. It has some basic redundant functions like carries thyroid hormone and vitamin A throughout the circulation. These are redundant functions, not really that important. But unfortunately, this protein is very unstable. It misfolds, it falls apart. And when it misfolds, it falls apart, whether due to mutations in the gene or just naturally. It's a naturally unstable protein. It causes amyloid, and it causes destruction of a lot of organs, primarily the peripheral nerves, causing a fatal peripheral polyneuropathy or cardiomyopathy. It builds up in the heart and causes heart failure. It could also affect the kidneys and so forth and so on. And nobody was working in this field. Nobody knew what this disease was at the time. When I gave a seminar, when we had the partnership with Eli Lilly that I touched on earlier, I was in Indianapolis, and I gave a seminar at Indiana University, and a fellow named Merrill Benson came up to me, a professor at Indiana, and he says, "This diabetes stuff, you're wasting your time. I got an application for your technology." And it was TTR. And I said, "Tell me more." What we did together, and this is work I did with Shu-Ling and many others, is that we developed transgenic mice that expressed the mutant TTR gene. We showed on the left bottom corner for the first time that you can actually lower TTR protein in transgenic mice. And then we took it into phase 1 development and showed for the first time that in normal volunteers we can lower transthyretin levels dose-dependently and very effectively. Now, I also want to mention here again that my ability to get this program into development at the time at Ionis, we had a lot of priorities and a lot of things to develop, but was very difficult. And when that has happened to me in my history, is I find someone else to help us do that, and I found a partner, as I've done many times before, a pharma partner. And that pharma partner supported us moving it into the clinic, and that's the phase 1 data there. So the business model at Ionis at that time was entirely dependent on partners for commercialization and late-stage development, pretty much most late-stage development. And then we initiated for the first time ever a phase 3 randomized clinical trial in TTR amyloidosis with polyneuropathy. I don't need to go through all these data. It's a randomized study where it was placebo-controlled, and the primary endpoint was neuropathy progression, something called mNIS+7, and it was a co-primary endpoint, and the other primary endpoint was quality of life, Norfolk quality of life, 15-month treatment period, and we were the first to demonstrate and validate the TTR lowering hypothesis that if you lower TTR, you can block progression of polyneuropathy in people with hereditary TTR amyloidosis, in which there were no effective treatments available at the time. On the left is the co-primary endpoint, peripheral polyneuropathy progression, mNIS+7. On the right is Norfolk quality of life, incredibly productive. Incredibly effective. And we were very, very proud to be the first to demonstrate this. And this drug, inotersen, brand name Tegsedi, was approved by the FDA on October 5th, 2018, published in the New England Journal of Medicine, and we were very proud of that. But this medicine was not a wildly commercial success. Why was that? Well, at the time, our partner helped us get the drug into clinical development and so forth, but then what happens sometimes when you have partnerships is that new leadership comes in, and they decided that they're out of rare diseases. After our NDA was actually under review by the FDA, and Ionis does not have a commercial organization to deliver the medicines to patients. Right? This drug had side effects, folks. I'm not going to minimize those. They had platelet effects, but they were extremely rare. And the profile of this drug was highly differentiating and should have helped a lot more patients than it did. But because we weren't able to launch the drug on time, it takes years to prepare for a commercial launch. We were starting from stop, and we had to fill some stopgaps and find someone else to commercialize it. It wasn't a commercial success. This was a non-GalNAc version of our TTR-lowering drug, was approved, but we persevered. We continued. Our drug discovery team, now I'm moving into kind of approaching my CEO role at Ionis, and we developed the GalNAc version of inotersen. It's called eplontersen. And I don't have to tell you how GalNAc works, except to say that in preclinical models, the potency was remarkable. Fast forward many years, we brought this drug into phase 3 development, and it was incredibly effective, even more effective than inotersen was. Shown in the middle here for eplontersen was the serum TTR reductions. There actually are error bars there, people. It is incredibly strong, powerful, tight reductions in TTR. On the left is the mNIS+7 that I already shared with you. You could actually see people are improving. It's going below the line, the zero, the axis. People are actually improving their disease, not just halting it. Same for Norfolk quality of life. People are getting better with eplontersen. And based on these results, another bench-to-bedside success, Wainua, eplontersen, was approved in December of 2023, and this was about my third year into being CEO. And I'm pleased to say that this medicine is doing very well, reaching many patients, thousands and thousands of patients, and helping them and changing their lives immensely. And I'll get to how we're delivering that medicine in a few minutes. Very proud of Wainua. There's another study that's ongoing with this same medicine that we prioritize. I pushed forward as CEO, and we moved into phase 3 development, and that's for the cardiomyopathy indication. And we initiated and are conducting today at Ionis the largest study ever conducted, a cardiovascular outcome trial for eplontersen in patients with TTR cardiomyopathy, heart failure. The study's fully enrolled, and we expect data in the second half of the year. And assuming the data comes out in time for next year's OTS, I really recommend to the organizers of next year's meeting to invite Sam Semikis and to present this data. This is groundbreaking landmark study, and we can't wait for the data to come out. I'll give another benches to bedside story. I'll bring you back to the factor 11 story, the coagulation factors that we worked on earlier, right? Intrinsic pathway, extrinsic pathway, common pathway. We were disappointed our factor 11 program didn't move forward. We were proud of the fact that we uncovered new biology for the world to see. And as I was working with my team, this was an exploratory research and drug discovery at the time, we asked the question, what else do these pathways do besides thrombosis? And what we uncovered, what we learned, was that there's a whole other pathway here that is directly linked to the intrinsic coagulation pathway. And in fact, is directly regulated by the factor 11 pathway, and it's called the prekallikrein bradykinin pathway, right? And this is work I did with Alexei Ravenko and Ram McLeod and many others. And what we developed was, and what we recognized or what we learned was that there's a disease called hereditary angioedema that is directly related to, in fact, it's caused by overactivation of the prekallikrein bradykinin pathway. That's what HAE attack means, right? So we said, well, let's pursue it. And Alexei developed models of hereditary angioedema, and the data looked fabulous. It looked fabulous. And we moved it into clinical development. But before I get there, what is hereditary angioedema? Very quickly, it's a rare, chronic, genetic disease that can be fatal, in desperate need for more effective prophylactic treatments to prevent these HAE attacks from happening. What are HAE attacks? They're massive swelling of tissues, shown on in this figure here, which can be fatal if it affects the throat. You suffocate. They're unpredictable. So you can be at work, you can be in this meeting, you could be at your daughter's wedding and have a severe attack. They're incredibly disfiguring, debilitating, and they start to affect people in their first few years of life. Can you imagine how terrible that can be? So we discovered donidalorsen, a GalNAc second generation chimeric gapmer oligonucleotide targeting prekallikrein. The preclinical data, as I mentioned, was outstanding, was excellent. We thought we had something here. And I don't need to go through the GalNAc pathway with you, but donidalorsen was highly potent and highly effective. And we started with, of course, phase 1 data, phase 2 data in HAE patients was phase 2. And what we learned, I remember exactly where I was. This was in the middle of the COVID pandemic. I was CEO. And I was sitting in my car, and Richard Geary called me up. And I looked at my phone, I said, please, let this be good news. And what Richard told me was the data, the phase 2 data in HAE was stunningly positive, like 90% reduction in HAE attacks in that phase 2 study. So we moved it, and now I'm in the CEO role, and we moved it into phase 3 development, and the results were equally stunning. We looked at every 8-week dosing, we looked at every 4-week dosing, every 8 weeks in orange, every 4 weeks in blue, reduction of HAE attacks in this study, which was greater than 80% in the randomized portion of the study. And then when we treat continuously in the open label extension, it reached almost complete elimination of HAE attacks, administered once per month or once every 2 months. It was incredible. This is work with Ken Newman and Lara Bordone on the clinical side. This data was so compelling that it was published multiple times in the New England Journal of Medicine, the phase 1 data, the phase 2 data, and the phase 3 data. And in August of this year, this medicine was approved by the FDA as a prophylactic treatment for hereditary angioedema. And this was, and I'm proud to say, I'm not taking credit, there's an amazing organization that allowed all this to happen. But I'm very proud to say that this was the 4th medicine, FDA-approved medicine, during my tenure as CEO. And the launch is off to a very, very good start, although it's in its early days now, but it's going very well. final chapter, a new job, a new plan. I want to tell you about the CEO leadership transition at Ionis, right? The board of directors and the founding CEO, Stan Crook, approached me in around 2016, 2017, I don't remember exactly, to move into the CEO role of Ionis. And there would be a 2-year transition period there, 2018 to 2020, and I would take the helm in 2020. And initially, I wasn't that thrilled about the idea. I'm an R&D guy, but I love Ionis, and I love what the potential this company can achieve, and I accepted. And during the transition period, I was asking myself questions. This guy again, I actually do that a lot in the hallways. People, like, when they see me do that, they run away scared. But I was asking myself, well, what's holding us back to reach our full potential? And why aren't the drugs that we conceive, discover, and sometimes develop, at least early on, making it to patients? I mean, it's awful, right? If a breakthrough treatment doesn't get to patients when you've invested so much in it and patients are depending on you. And where the hell did all this competition come from? I showed you the I showed you the companies that were founded in 1989, and they're all essentially gone. And now there's a whole ton of them. And Joe Scalzo said in the opening video, 150 oligonucleotide companies today. Well, these are the questions I wanted to address, and I was going to address them in 3 ways, 3 strategic imperatives. Stop relying on partners. I told you stories that broke many people's hearts. For the drugs that we conceive, discover, not reaching patients, we're going to build our own pipeline. We're not going to partner it. We're going to prioritize the wholly owned Ionis pipeline, and we're going to build an innovative, highly experienced, excellent commercial organization to match the excellence we've always had in research and in development, and deliver those medicines to patients ourselves. And for the competition and all the great work that's being done, we build off all the exciting science that the whole world is creating, expand and diversify our drug discovery capabilities, medicinal chemistry, new mechanisms of action, new strategies, and so on. And I'm pleased to say that this plan is going exceptionally well. 2020, I moved into the CEO role. For those that are aware, there's a conference called JP Morgan Healthcare Conference the first week or 2 of January, and there I was, my inaugural JP Morgan conference, new CEO, and I laid out a plan, and nobody believed me. Ionis is an R&D organization for 3 decades. Nobody believed me. But that was okay. I laid out a plan, that new strategy, and I wanted to do this in a measured, careful manner. Not get over our skis. We're going to focus on the U.S. market initially and find partners for ex-U.S. commercialization. I should get board approval at some point, which I eventually did, and prioritize the pipeline. We're going to start with a co-commercialization relationship to help build our commercial organization under us to prepare for our first independent launches for the company. And again, this is all in the interest of patients. ApoC3 was the program that I prioritized for our wholly owned pipeline that was going to get everything it needed to get to bring it forward, right? You heard from Dr. Alan Brown yesterday, an outstanding presentation on how ApoC3 affects lipids, triglycerides, in the management of diseases associated with severely elevated triglycerides. You're going to hear a lot more about that tomorrow from Sam Semikis in the clinical presentation section. The target is ApoC3 for the management of triglycerides. So I don't need to go into the mechanism of this. We've already gone through that. And I really don't need to go into the first indication we pursued, which was familial chylomicronemia syndrome, FCS, a fatal genetic rare disease that Dr. Brown took you through yesterday morning in elegant fashion. But basically, these patients have super high levels of triglycerides, 10, 100 times normal levels. They have all kinds of problems, including a potential of fatal, the risk of acute pancreatitis, which is often can be fatal. And if it's not fatal, it ends up putting people in the hospital for weeks at a time. We had a lot of experience with FCS from a prior drug, non-GalNAc version of the drug that I'm about to tell you about. It's called volanesorsen. So I don't take credit for the great science that people did at Ionis before I was the CEO in validating for the first time ApoC3 for FCS. But what I did do was prioritize a better version that I thought would get approved by the FDA, the GalNAc version, which is olezarsen. And you saw this data yesterday presented by Alan Brown in patients with familial chylomicronemia syndrome, phase 3 data. These are the ladies and gentlemen that played a pivotal role in the development of our ApoC3 program at Ionis. Shown here is the primary endpoint, triglyceride lowering, that Dr. Brown showed yesterday. But not only did we show substantial reductions in triglycerides that got many of these patients near normal or at normal, we showed that we can reduce acute pancreatitis in these patients and reduce hospitalizations and improve quality of life and so on. It was really remarkable. So as this program was advancing forward, our first top priority wholly owned program for Ionis to bring forward ourselves, very importantly, Tryngolza was approved in December of last year for the first ever FDA-approved medicine for familial chylomicronemia syndrome. And we were incredibly proud of that. And this is the first medicine that Ionis has taken to patients directly, commercialization ourselves, and we moved fast. And that's one of the advantages of having your own organization, is to be able, you can move with urgency and deliver these medicines quickly to patients. And the stories we're hearing back from the patient community are just amazing, how well these patients are doing. As we were developing the phase 3 program for FCS, we were asking other questions, and me and my teammates at Ionis were wrestling with what's the next indication. There's a lot of diseases associated with severely elevated triglycerides, and we were talking about diseases like MCS and persistent chylomicronemia. We were even talking about doing a cardiovascular outcome trial potentially in this area. And what we settled on after months and months of research into the unmet need and the potential for us to be able to deliver, we settled on a disease called severe hypertriglyceridemia, which is very similar to FCS. It's just doesn't have a direct genetic component cause of the disease. But what it does have is symptoms and comorbidities very similar to FCS, including the risk of a potentially fatal acute pancreatitis attack. And there are no effective treatments for SHTG today. They're treated with fibrates and omega-3 fatty acids. They're not effective. They don't work well at all. And this is not a rare disease. Millions of people in the United States suffer from severe hypertriglyceridemia, SHTG. So after phase 1 development, some preliminary work in phase 2, we initiated the phase 3 program just a few years ago. Olezarsen, the GalNAc version of volanesorsen. And we conducted 2 randomized clinical trials to support FDA requirements, also a supportive safety study. The studies are called CORE and CORE2. The phase 3 results were, the design is shown here. The primary endpoint was triglyceride lowering, and the key secondary endpoint was acute pancreatitis, just like FCS. In September of this year, we announced groundbreaking results in this population. On top of standard of care, we showed that we can lower triglycerides by an additional 70% plus, getting many patients into the normal range. Right? I forgot to mention, SHTG is defined by triglycerides 500 and above. Normal triglycerides is 150 and below. Most of these patients are in the thousands. We normalized triglycerides in many of these patients and got them out of the SHTG range and many more. Not only that, we were for the first time ever in SHTG showed statistically significant reduction and robust reductions in acute pancreatitis, 85% reduction. We essentially prevented AP from happening with good safety and tolerability. The sNDA, supplemental NDA following FCS, will be in by the end of the year. We expect FDA approval in the second half of next year. And we're really proud to have been chosen to present this data at the late breaker clinical presentation at the American Heart Association on November 8th, a very prestigious session at AHA. So the path to controlling our own destiny, our own independent launches, to making sure the drugs that we conceive, discover, and develop make it to patients is going great. I mentioned that I wanted to start with a co-commercialization partnership. I talked to you about Wainua, eplontersen. That partnership is with AstraZeneca, and that co-commercialization partnership is doing great. And as I mentioned before, we got a lot of happy patients on Wainua today. It's doing very, very, very well, as is Tryngolza for FCS. Donzera, our second independent commercial launch for HAE, is doing very well. And next year we expect 2 new FDA approvals, SHTG, and a drug for a rare neurological disease called Alexander disease, which we shared. We announced positive phase 3 data in September also. September was a great month for Ionis, and a lot more coming after that. So to wrap up this last section, I want to bring you back to the plan that I laid out in 2020. I already took you through at a high level prioritization of that pipeline for Ionis. We're the top priority, not partners. And to build a innovative commercial organization to deliver the medicines that we conceive, discover, and develop directly to patients. If you walk away from this presentation thinking that, oh, Ionis went commercial, you missed the message. That would be deeply disappointing to me. The science at Ionis has always been our strength. Science is our foundation. Innovation in research, innovation in clinical development has always been what differentiates Ionis from many others. And that is still the focus for the company today. Under my leadership, under previous leadership, and for any future leader that, if I have any say who that would be, under that leadership as well. Science is king at Ionis. We just happen to now deliver drugs to patients that we bring forward. But in the realm of science, I want to touch on, in the last few moments, the last pillar, expanding and diversifying our drug discovery capabilities. I remember coming back from that JP Morgan healthcare conference and asking Eric Swayze to pull together the research organization and to talk to them about how we're going to do things a little different going forward, that we're going to invest in many new areas in research in 2020, focus on 3 key areas. We're going to expand our technology platforms. In addition to antisense, we're going to expand it to new platforms. We're going to strengthen our ability to target tissues and cell types outside of the liver, right? While strengthening our liver programs of TARS-CRU, and to use all that to strengthen and expand our therapeutic opportunities. So let me just take you at a very high level how that's going. Expanding our technology platforms. Ionis engineered using the great minds in our chemistry teams and our drug discovery teams are now delivering first siRNAs. They're in the clinic today, right? To complement what we're doing in the single-stranded space, we're using double-stranded spaces for applications where it makes more sense to do that. And you're going to see some of our first clinical data tomorrow from Sam Semikis, phase 1 data for ION 775 that supports semi-annual or maybe even annual dosing. New chemistries like NMA. We're leading the way in CNS diseases using intrathecal routes of delivery. However, the investments we're making in chemistry are now allowing us to dose once per year intrathecally, like salanersen, which is a follow-on molecule discovered by Ionis that is moving into phase 3 development with once per year intrathecal dosing based on the stability that Ionis scientists provide. And we're going to do the same thing for another splicing oligo called Dravet syndrome that start clinical testing next year. Other chemistries and gene editing. Using Ionis know-how in medicinal chemistry and everything else we know, applying it to gene editing is going extremely well. We launched this a few years ago, and we're expecting our first gene editing development candidate to be approved by the end of this year, moving into preclinical IND supporting studies next year. Targeted delivery is going great. We're focused initially on cardiac and skeletal muscle. You've heard a lot about this at the meeting. The first SI targeting cardiac myocytes is advancing into clinical testing this year, right? You heard a lot about this program from Adam Mullick in yesterday's presentation. We're expecting a second one. The first one is with a partner. The second one that was in a previous relationship we had with a partnership. The second one is wholly owned by Ionis. And we're making great strides in further strength. And this is allowing us, in skeletal muscle, allowing us to strengthen our work in neurological diseases by moving into neuromuscular diseases. And we're expecting 3 or 4 candidates in neuromuscular diseases next year in blood-brain barrier. To allow us to dose our drugs using low molecular weight conjugates attached to ASOs or siRNAs delivered subcutaneously. And the preclinical data in non-human primates is just amazing. And we're expecting our first candidate there for blood-brain barrier by the end of the year as well. And all of this supports our mission to expand our therapeutic opportunities, to strengthen cardiovascular diseases at Ionis, our leadership in that space, strengthen our neurology franchise with BBB and neuromuscular diseases, and even move into some new spaces like in pulmonary diseases. So now, to close, I can't say how much I appreciate all of the great colleagues and support, personal, family, as well as colleagues at Ionis and in academia, and many of the people out here today that have allowed me to have a little bit of success in my career. I also want to emphasize how much I really love this society. The OTS is a great society, as was mentioned in the opening video. We started in a castle with a dozen or so people. And we felt that the oligonucleotide area needs more attention. And we created the OTS, and I've been heavily involved in this, and I think we did good things, Masad, don't you think? And we got a lot done in those days, and it continues to be a great society. I am so appreciative of receiving this award from the OTS. Ionis employees, companies are made of people. People that are driven with passion and a commitment in our space to deliver life-changing medicines to patients. And the employees at Ionis are second to none. And it's been an honor and a privilege for me to be part of that community and to now lead it and to work with our community, our patient community too. This is an example of a recent Surf Away event we did in California, in which we bring in patients and parents and healthcare providers and Ionis employees that are volunteering their time to work with these individuals. It's an amazing organization that I'm incredibly indebted to. I also want to acknowledge an outstanding executive team that I helped shape to some extent during when I moved into the CEO role. Folks, we had some hard times in the first few years of my CEO. Richard remembers we had several late-stage disappointments. We had the COVID pandemic and so forth and so on. These folks have been behind me all along the way, and I would not be as successful as I am today without these folks. I've also had the privilege of 5 outstanding colleagues and people that have taught me so much. Dave Ecker, who hired me back in 1989. Frank Bennett, who was my boss for decades, and taught me so much about research. Richard Geary, who taught me everything I know about clinical development, preclinical development, regulatory affairs. Eric Swayze, an outstanding medicinal chemist and culture champion at Ionis. And of course, Stan Kruk, my PhD mentor, and the person who gave Dave Ecker the permission to hire me back in 1989 to come to Ionis. I learned so much from Stan, great leader, and I learned what we think Ionis should do based on what I saw in Stan and what I think we should need to do that's different. But I'm so indebted to him, of course. And then lastly, well, not lastly, my board of directors, which has been by my side and supportive of me right from the outset. I could tell you that I got a few funny looks when I came back and I said, hey, we're going to make Ionis into a fully integrated biotech company. And they looked at me and they said, well, what do you know about drug commercialization? Well, they understood that how I was going to do it. I had a plan, and they've been by my side and behind me all along the way. Now, finally, and most importantly, is my family. starting with my wife, Joanne, who's sitting here in the front row. We were high school sweethearts. We were actually junior high school sweethearts, and that's us in high school on the left side, back when I was a tree hugger. I had a Volkswagen camper, hiking, backpacking, all that stuff. I'm not that anymore. That's us in a more recent picture. And my children, Patricia and Jake, who inspire me every day. My brothers, Jim and Paul, who always are there by my side and helping me get through tough times and so on. And 3 guys from Jersey. I mean, you can't go wrong. My dog Martini, who sits with me and has a martini with me every night, whether he wants one or not. And of course, my parents, Jim and Ro, who are passed now. But they saw something in me that I never saw, and they stuck with me. And if I were them, I don't know if I would have stuck with me with all the trouble I used to get into in high school and so on. But I'm obviously deeply indebted to them. So now to finish, this is usually the time in these presentations that a presenter might leave you with a quote from a philosopher or a famous scientist or something like that. So I figured I would do the same. I'm going to leave you with this. Today's presentation was about change and embracing change and the importance of that, as well as to do things that you have never done before if you really want to create something special like Ionis. I couldn't find anyone to make this statement, so I made it up myself. This is my quote. But if you really want to create something special, folks, you got to embrace change and you got to do things you never did before. So with that, thank you very much. I really appreciate it, and I'll happy to take any questions if there are any.
Thank you, Brett.
Thank you. Appreciate it. Thanks a lot, buddy.
We'll get you a plaque here in a minute. I'm going to ask if there are any questions. We have about, thank you. We have about 1 minute for some questions. If anybody has a question, please step up to the microphone. We have one right over there.
Klaus Waltz, the University of Miami. Brett, that's fantastic. And you and I have been in this space exactly as long, I think. So we did some early work in CNS in the 90s, and we took your gapmer design, and did work with LNA in the ends and so forth back in the 90s. But lots of successes, but I could sense some disappointment around factor 11, right? So just pick that one. There are multiple modalities today, including siRNAs, right, for multiple companies. So what's the sweet spot? What's the winning drug, you think, at the end of the day?
I don't know, Klaus, and thanks for all that. What I do know is that it's really important that for antithrombotic effectiveness, you need to have consistent, durable lowering of thrombosis risk. You cannot have peaks and troughs. You cannot have peaks and troughs because when you lose a little bit of activity, you're at risk for a heart attack, right, or a stroke. And I'm concerned that small molecules don't have that durability.
Right.
So my bet would be on antibodies or oligonucleotides, if we can get them back in there, because they're so long-lasting and they clamp down the antithrombotic effects. So my money would be on antibodies at this stage.
How much of a knockdown before you get hemophilia B or something?
What we showed in our clinical studies was about 80% is enough.
Yeah.
But even in patients where we achieved 90, 95%, individuals, we weren't seeing bleeding.
Right.
So I think you have a nice therapeutic range there that you can tap into.
Great. Thank you.
Thanks, Klaus.
One more. Yes.
Thank you very much. A really nice presentation. This is my first OTS event, and I'm relatively new to the oligo space, so it was really nice to just see the overview of, Ionis and how this event came together. It was really interesting to see that your particular plan for Ionis and how you've started to, move into new areas like of commercialization, things like you say, especially with regards to the prevalent disease that you mentioned. Could you give any comment on, maybe for Ionis or the field more generally, how you see oligos moving into more prevalent diseases?
Yeah.
Is that the future, or do you think that's still a long way off?
rare diseases, I don't believe, have been traditionally pursued with oligonucleotide therapeutics because they're rare. It's because they're commonly caused by genetics. Genetic causes of rare diseases are generally well understood. And our technologies are, of course, precision technologies that can target the root cause of a disease, like TTR, for example, in transthyretin amyloidosis, or SOD1 in SOD1 ALS, and that kind of thing. Usually the cause of more prevalent diseases isn't as clear. The work we did on ApoC3 was really, we pulled a rabbit out of the hat. Sam Samikas will tell you about this. Nobody thought, actually, Dr. Brown mentioned it yesterday, nobody thought it was going to work, but it's much more risky. So you don't know, it's not as easy to find the right target for prevalent diseases. With that said, with the cracking of new areas like CNS diseases, as well as cardiovascular diseases, we're on the brink of knocking down the door for highly prevalent diseases. In cardiovascular, we're expecting pelacarsen phase 3 data in the first half of next year for Lp(a)-driven cardiovascular disease. Millions of people suffer from that disease, and I'll just stop there with cardio. In neurology, targeting tau for Alzheimer's disease, we've shown remarkable data in phase 2 showing improvement in cognition and reversal of pathology. I think we're on the brink of all these big diseases, so it's not just rare, but rare we started there for a reason, and that's it's understanding what the cause of the disease is.
Thank you so much.
Thank you. Thank you, Brett. Congratulations. Thanks. On a great career, and it's only starting.
I hope so.
There we go. All right, thank you.